Next up is a discussion of cells, the foundation of life, the basic building block of every living thing are cells. Cells are individual. Human beings have millions and billions of cells. There are some types of organisms that only have a single cell, like for example a bacterium. A bacterium, which make up bacteria, which is the plural of bacterium, contains only a single cell. But every living thing is fundamentally made up of cells. The two basic types of cells are prokaryotes and eukaryotes. Prokaryotes are cells that are undeveloped. Eukaryotes are cells that are more developed. Basically, very, very basic organisms like bacteria are prokaryotes, and eukaryotes are things like animal and plant cells. Now, the main difference is that eukaryotes, the bottom one, the more advanced one, have a defined nucleus in the middle. A nucleus is the thing that has the DNA, and the DNA is the thing that really runs the cell. We're going to look at some examples as we move along in the chapter, but this is an example of how a cell generally looks. You've got the cell membrane all the way on the outside. We're going to look at the various organelles, which are these things in the middle. But in the very middle over here, you have what's called the nucleus. And the nucleus is where you have the DNA. On the other hand, when you're talking about a prokaryote, which means before, pro always means before, you've got something like this, a prokaryote cell. It's similar in terms of the basic structure. This one happens to have a cell wall, but that doesn't matter. What does matter is there's no defined nucleus. The DNA, this orange stuff in the middle over here, isn't, whereas before we saw the DNA would be somewhere over here in this area, that would have a defined nucleus in the middle. A prokaryote cell over here would not have a defined nucleus. Instead, the DNA would just kind of be swimming around in the middle of the cell. Okay, so the prokaryotes are in bacteria, things that are very, very not advanced, things that are very primitive. Whereas eukaryotes are things like plant and animal cells. They have a real nucleus, and we're going to discuss a little bit later on the distinction between the plant cell and the animal cell, but certainly there are many very important distinctions between them. Now, there's basically a distinction between animal cells and plant cells that we'll look at in a little bit, but first I want to look at the various things that are inside a cell. A cell's got lots of organelles in the middle. This is just an example of, well, this happens to be an animal cell. But you can see in this one, and in the one that we looked at a little while ago, that there are many different materials inside the cell, and those are called organelles, kind of little organs. You've got the lysosomes. The lysosomes contain the digestive enzymes. In other words, they break down food. They break down polysaccharides into simple sugars, big amino acids into individual proteins, big fat chains into individual lipids, the sort of thing that are necessary for the cell to break down whatever nutrients are necessary. And they also break down defective organelles. If some of the mitochondria or whatever it is aren't working, they can be broken down and recycled, or at least gotten rid of through the waste process in the body. You've got the membrane. The membrane is the outside of the cell, kind of the thing that stops materials that are supposed to go in, or that are supposed to stay in from leaving and supposed to stay out from coming in. It's made of phospholipids, which are fats with phosphorus. That's why it's called phospholipids. Normally, lipids only contain carbon and hydrogen, but there are phosphorus molecules involved as well in the plasma, in the membrane, and proteins in the membrane as well. The job of the proteins is to communicate, really to facilitate the communication between the cell and the environment. In English, without going through, you can read this on your own, obviously. But basically, what the membrane does is specially structured and specially engineered, not just to keep things in or out, but also to allow transport one to another. Sometimes things have to leave the cells if the cell wants to get rid of it or transport it. And then there are some things that have to go into the cells if the cell needs the materials, sugars, proteins, whatever the cell needs. Cell membranes look something like this. You see, you've got the lipids, you've got picture this as being inside the cell, and up here as being outside the cell. And you've got this structure, the main structure all over here, are fat cells that are blocking things from coming in and blocking out and coming out. But you've also got protein molecules, protein channels that kind of open and close to allow things necessary to move in and move out. And those are the things that actually control ingress and egress from the cell, which means moving in and out of the cell. That's, of course, the function of the membrane. Some other things, we've got vacuoles and vesicles, which transport and store also materials. If there's water or food that the cell needs and it's going to keep for a later period of time, that is what is known as a vacuole or a vesicle. The ribosomes produce the protein for the cell, take the amino acids that are imported by eating or whatever other process the cell gets it, and then go ahead and produce whatever proteins are necessary for the cell. We learned last time all about the different types of proteins that you have, things like hormones and enzymes and materials that move information in and out of the nucleus. These are all things that are necessary that are produced by the ribosomes. You have the endoplasmic reticulum, or the endoplasmic reticulum, excuse me, the ER, which synthesizes membrane components and allows things to travel throughout the cell. We're going to look at a diagram in a minute. The Golgi apparatus, or the Golgi bodies, packages and sorts proteins and lipids, kind of the manufacturing plant, or really the packaging plant. The manufacturing plant would be the ribosomes, and then the Golgi apparatus would be the packaging plant, where it puts everything together and moves it around through the endoplasmic reticulum in terms of where it has to go. Other things that could be involved in certain cells, you've got cilia. Cilia are kind of like hair-like structures, and flagella are longer, whip-like structures that allow the cell to move around. The cilia kind of allows the cell to feel things, the flagella to actually move things. Some certain types of bacteria or certain types of protozoans, which are tiny, tiny, tiny little organisms that live in water, including in our bodies and in our mouths and whatnot, travel around through the flagella. The nucleus contains the DNA, contains the genetic material in the inside. The chromosomes are they make up the DNA. DNA is made up of chromosomes. The chromosomes are the thing that essentially tells our body what to be. The reason why you are a human being and not a fruit fly, I mean, aside from everything else, is because of the fact that your chromosomes indicated that you should grow a brain and grow eyes and grow whatever, and the fruit fly only indicated that it should grow a small little set of wings and whatnot. That doesn't mean that you're in danger of turning into a fruit fly if your DNA changes a little bit. Our DNA is quite a bit different from the fruit fly's DNA, but the fact of the matter is that the chromosomes are kind of the blueprints on which the cells know what to do, and the cells go out and do their jobs because those jobs are written in the blueprints. The nucleolus is the kind of the protein synthesis hub inside of the nucleus. The nucleus itself in the middle has the nucleolus in the middle, which is where the proteins are generated. Essentially, the nucleolus, it's kind of complex. I don't want to go into it right now, but the chromosomes, namely the DNA, have to be able to send its message throughout the cell, and the materials by which that message is sent are produced in the nucleolus. The nuclear envelope covers the nucleus, just like the cell membrane covers the cell itself. You have the mitochondria. The mitochondria does the energy production, the respiration. The mitochondria is the thing that takes the sugars that we eat or that we have and generates energy. It does it by a fairly complex process called respiration, either by aerobic respiration, anaerobic respiration. I don't want to get into that right now. But the mitochondria are the thing that create something called ATPs, adenosine triphosphates, which essentially are able to transport energy. It's able to store energy in these ATPs. The ATPs can go wherever they're needed and release the energy that are necessary. Then you have the centrioles. The centrioles are necessary as part of the reproductive process when one cell splits apart into two cells in a process called mitosis. And then you have the cytoskeleton, which really just forms the more solid parts of the cell. Things like flagella, cilia, things like that. These are all different, and this is not even an exhaustive list. There are plenty of other things that may appear in an individual cell. And like it says over here, don't necessarily memorize the list, but you should be able to distinguish between different types of cells and the functions of what some of these things do. A plant cell is slightly different than an animal cell, in that except that they have a cell wall in addition to a cell membrane. They also have big plastids they kind of look like very big vacuoles, very big storage areas. They have chloroplasts, which are necessary for photosynthesis. Photosynthesis means making food. A plant makes its own food, whereas a human being doesn't make its own food. I mean, it can cook its own food, but it doesn't actually spontaneously generate its own food. It has a central vacuole, a much bigger vacuole than an animal cell. Plant cells don't have lysosomes. Flagella and cilia are very often involved in movement, and plant cells, of course, don't move, so they don't need that sort of thing. Here's an example of a side-by-side comparison. I have some diagrams that'll be a little bit easier to show you, but just as a general principle, you can see some of the distinctions. The plant cell's got a cell wall, or the animal cell doesn't. The animal cell is more circular. The plant cell is more square. The plant cell's got a much bigger vacuole over here in the middle that stores a lot of food and stores a lot of water and whatnot. But they both have nuclei, they both have mitochondria, they both have a lot of the other things that are necessary. Here are some other examples. Here is an animal cell fairly round, and it has a nucleus in the middle with the nucleolus in the middle of the nucleus, lysosomes, and a plasmic reticulum, which is kind of the transport mechanism from the nucleus to other parts of the cell, the Golgi apparatus, the mitochondria, which remember do the respiration for the cell. Then here's an example of a plant cell. Plant cell looks again fairly similar. You have the cell wall outside the cell membrane. You've got the Golgi apparatuses, just like I guess, just like you do in the animal cell. You've got chloroplasts. These do the photosynthesis, which you do not have in the animal cell. You've got the nucleus and the nucleolus in the middle over here. You've also got a huge vacuole that takes up a lot of the cell. The last half of this chapter goes more in depth into the cell membrane and materials moving from inside to outside and from outside to inside of the cell. Every cell, whether a plant or an animal cell, has a cell membrane, which is an outer layer that is surrounding the cell. Its main function is to keep things in that are supposed to be in and keep things out that are supposed to be out, but also, of course, to allow things in that are supposed to come in and to allow things out that are supposed to come out. They regulate biochemical reactions by allowing the ingredients for those biochemical reactions in and out of the cell. They separate the organelles from the surrounding interstitial fluid, which means the fluid that all the cells are kind of bathed in. You know, all the cells in the body, let's say in particular in the skin or in your heart or whatever it is, are kind of bathed in this fluid that where all the cells sit in. Well, you want to stop or prevent that material from interfering with the organelles. It also separates between the membrane that are enclosed within the organelles within the cell and the material outside the cell. It also creates an attachment in between cells by latching onto other cells, hooking up with other cells, moving next to other cells, and allowing things to be moving back and forth. That's really the last thing, to regulate the exchange of substances, to make sure that the material inside the cell can get out, and the material outside the cell can get in. And a lot of the movement between inside the cell and outside the cell, and between different cells, is based on the materials being dissolved in water, and the water moving from inside the cell to outside the cell. Obviously, it's a lot easier for the cell to move water in and out, because water is very, very small and very permeable. It moves into materials a lot easier than solid materials. Now, that doesn't mean that solid materials can't move from outside the cell to inside the cell. They can, but it's a lot easier when they're dissolved in water. A substance that can be dissolved, for example like salt, is a solute, whereas the liquid in which it dissolves, in this example of coarse water, is the solvent. So when you have salt water, the salt is the solute and the water is the solvent. Concentration is the amount of solute that is dissolved in the solvent. You could have salt water that is very concentrated, or you could have salt water that is not concentrated. Salt water could be a little salty, or salt water can be extremely salty. So concentration of the solute is a measure of volume. For example, just a very simple example, if you have 2 cups of water and you've got 5 tablespoons of sugar in one and 3 tablespoons of sugar dissolved in the other, obviously the first cup has a higher concentration than the second. If you were measuring the amount of material that is dissolved, the first cup you would say 5 tablespoons per cup, and the second one you would say 3 tablespoons per cup. Very simple. Obviously that's a 5-to-3 ratio, and the first one has the higher volume. Now, the thing about materials moving inside the cell and outside the cell is that water is always drawn towards an area of higher concentration. Salt draws the water towards it. So that's why, for example, when you salt, if you want to get rid of the blood in a piece of chicken, you can salt it. You can put the salt in on it, and the salt kind of draws the water out of it. That's why salting a chicken works to get rid of the blood. And salting anything works to draw the liquid out of it. Water is drawn from area to an area where it's a higher concentration. So, for example, if you have salt water outside the cell and non-salt water, fresh water inside the cell, the water from the fresh water is going to be drawn into the outside. It's going to be drawn to the area of where there's more concentration, where there's more salt on the outside. Materials themselves, like the salt itself, tends to move from areas of high concentration to areas of low concentration. If you, for example, have very heavily diluted salt water, very salty salt water, and a very not diluted salt water, very not salty, watery salt water, and you pour from one to the other, of course the salt is kind of going to spread out and even itself out so that eventually the entire thing is going to be pretty uniform. The entire cup is going to be pretty much the same. And generally speaking, whenever you have something dissolved in water, the material, the sugar, the salt, the solute, whatever is dissolved in it, is going to move from an area of high concentration, where there's a lot of it, to an area of low concentration. That's why eventually everything is going to even out. That's a very important concept in the concept of things moving into and out of a cell, because whenever you have a higher concentration outside the cell, whatever it is in higher concentration, it's going to tend to try to move through the cell membrane into the cell. Gradient is how you measure the difference in certain properties. If you have highly concentrated inside and lower concentrated on the outside, there is a gradient, which means a distinction between the two. In that example we discussed before, when you had 2 cups on one, 5 tablespoons per cup on one side and 3 tablespoons cup on the other side, so there's a drop-off if you move in between those two regions. If you move in between those 2 cups, you're going to have a drop-off in concentration from 5 to 3. That's called gradient. Gradient is also sometimes used when you're talking about, hills, a distance in elevation. A gradient, if you have a steep hill, you're going from a higher elevation to a lower elevation. That's also called a gradient. So in this context, it means a difference in concentration. Now, a cell membrane is selectively permeable. Permeable means something can infiltrate it, something can move into it and out of it, and selectively means only some things. Not everything can move in and out of the cell membrane, but on the other hand, it's not completely airtight or watertight, which means some things can move into and out. All those concepts really brings on our next issue, and that's the issue of how things get transported into and out of the cell, and whether energy is necessary for that process. The 2 types of transport of which materials can move into and out of a cell are passive transport and active transport. Passive transport does not require energy, whereas active transport does require energy. And to visualize this, I know it may seem very complex in the abstract. I want you to visualize, instead of the cell membrane being a cell membrane, visualize it being a paper towel. And you've got liquid on the outside of the paper towel, and you've got liquid on the inside of the paper towel. Now, yes, if it were a paper towel, it would tear fairly easily, but imagine it's the kind of paper towel that doesn't tear. Maybe it's Bounty, according to their Bounty commercials. So we're going to think about it that way in terms of something being selectively permeable, where not everything can move in and out, but some material can move in and out of beyond the paper towel. Passive transport is the sort of thing that happens automatically, either because the concentration is lower inside than it is outside, or because it's water which can just kind of slide through the walls. Whereas active transport, where you have a big molecule that cannot so easily go between the inside of the cell and outside of the cell, it requires energy in order to move it from one place to another, and that is what you would call active transport. So let's take a look at what would constitute passive transport. Passive transport is also sometimes known as diffusion. Okay, let me have the wrong slide up, and now I'll put the correct slide up. Anyway, so diffusion of substances happens across a membrane down a gradient of concentration, pressure, or electrical charge, and don't require energy. Diffusion, or passive transport, occurs either when you're going from a region of high concentration to low concentration, like the salt moving from salty salt water to non-salty salt water, or to regular water. That happens automatically. It happens naturally, because things tend to move from areas of high concentration, where there's a lot of it, to areas of low concentration, where there's not that much to it. So that does not require energy. Pressure. Things tend to move from areas of high pressure to areas of low pressure, just like wind. Wind is where you have high air pressure in one area and low air pressure in the other, and the high air pressure blows to the area of low pressure. And electrical charge, which we'll get to in a little while. Electrical charge can also naturally allow things to move from one area to another. The types of passive transport that we're going to look at are simple diffusion, which is a diffusion of either water, which naturally moves from place to place, like through a paper towel, lipid-soluble molecules, or gases, dissolved through the membrane. In other words, things that are so small or so dissolved in the water that they can kind of squeeze in between these fatty materials over here. As you can see, these fatty materials on the outside of the membrane, if you have water over here, it just kind of squeezes through the little holes and goes inside the cell. Or if you have material that is concentrated, or a material that is dissolved in the water, it can just kind of move through without any help from these big red things that allow certain things in and out. That's one. The other type is facilitated diffusion, which is water or water-soluble molecules through a membrane via a channel or carrier protein. That's something that goes through these red proteins over here, transmembrane proteins, where the proteins kind of, as we looked at last time, kind of open up a little bit and allow things that wouldn't ordinarily be able to pass through these fatty substances, either because the materials are too big, they're not dissolved enough, and they need the membrane to kind of open up, and that's what these proteins do to allow them in. They still could be passive transport, because remember, if they're coming from an area of high concentration up here to an area of low concentration down here, or high pressure up here to low pressure down there, they still don't require energy, but they still need something to open up and allow them in, and that's the purpose of these membranes. And then we have osmosis, also a form of diffusion, also a form of passive transport, which is diffusion of water across a selectively permeable membrane, like the ones on the outside of cell, from an area of higher free water concentration to an area of lower free water concentration. In other words, there's room for more water to come in, and that is what is sometimes known as osmosis. A couple of other things about passive transport, about diffusion. First of all, it occurs when the temperature is higher. When particles of sugar are moving from place to place, again, the example is pretty obvious, it'll diffuse faster at a higher temperature at 75 Celsius than only at 45 degrees Celsius. In general, things that are hotter, environments that are hotter, are more conducive to dissolving things, are more conducive to chemical reactions in various different ways, in many, many different contexts. One example you might realize is that from your own experience, you probably know that things tend to dissolve faster in hotter liquid than in colder liquid. If you're putting sugar sugar in your coffee. If your coffee is cold, it's going to take a lot of stirring, and even so, you might not get all of the sugar to dissolve. On the other hand, if your coffee is piping hot and your coffee is boiling and you put sugar in, it's going to dissolve pretty much 100% and pretty much right away. The greater the concentration gradient, the faster diffusion will occur. In other words, the more the concentration difference, the faster things will move. If you have very salty salt water and you put it next to you mix it in with non-salty salt water, you put it in, like, let's say, a paper towel in between, it'll move very quickly. Whereas if the concentrations are pretty close, it may not move as quickly from one side to another. Things tend to even out as quickly as possible, so the higher the gradient, which means the higher the difference in concentration between the two sides, the faster one will move to the other. Example, you've got particles of sugar being diffused into two cells. Inside the first cell, the concentration is 25, and the second is 35. The gradient is relatively light. Well, forget the gradient between the two. Both cells are in a solution that has a 60% concentration. So the gradient between the 60 and the first cell is 60 to 25, whereas the gradient between the solution and the second cell is 60 to 35, which means the sugar water, the one with the sugar will move from the solution, which has the higher concentration, to the cells, but it will move faster to the 25% cell because the gradient is more, the difference in concentration is more, than the other one, which is the 35% cell. Osmosis works very similarly. Remember, osmosis is really just a form of diffusion when you're talking about water and water with a concentration of materials with a solution where the materials are dissolved in the water. And so this is, again, where you have water across a selectively permeable membrane, like a cell membrane, from an area where there's more water to an area where there's less water. So when and again, all of these things that we're going to discuss now are really just corollaries to what we discussed. There shouldn't be any major surprises over the next couple of slides. The more solute a solution has, namely the more concentrated it is, the less free water there is. Therefore, there's going to be, the more empty the water is, the faster the water with solution, the faster the water with dissolved particles will move towards it. Hypertonic means a solution with a greater concentration than the solute, and hypotonic is the opposite. Hypo means less, hyper means more, generally speaking. And so hypotonic means with more, it's just a terminology. Hypotonic is a solution that is more diluted, which means it is less concentrated. So just to give you an example, if you have a hypertonic solution next to a hypotonic solution, that means you have a more concentrated solution next to a less concentrated solution. The solutes, the sugar, the salt, whatever is dissolved, will move from the hypertonic solution to the hypotonic solution. Again, just to reiterate, hypo means less, hyper means more. Hyperthermia is when you're too hot. Hypothermia is when you're too cold. Isotonic is when the two things are equal. You have two solutions that are next to each other, separated by this membrane, by this paper towel, whatever it is, and they have either concentrate. Can you guess about what's going to happen in terms of movement? You can pause it and think about it. Okay, now that you've unpaused it, the answer, of course, is that there is not going to be much movement between one and the other because their concentration is equal. So water moves by osmosis from the hypertonic solution, which means the more concentrated solution, to the hypotonic solution, which is the less concentration solution. Here's an example, again, extremely obvious. It allows water to pass, but it doesn't allow salt water to pass, but the cell contains a 3% salt solution. The cell is then placed in a 5%, so obviously water will move out in the cell because it is going to try to equalize the amount of the concentration overall. If, for example, the cell and the water outside the cell was the same amount of material, eventually there would be 4% concentration across the board. That's also why water tends to be drawn out of salt water, as we discussed earlier, because the salt is drawing the free water in order to equalize the concentration. A osmosis across a plasma membrane is absolutely critical for all living things. All plant cells, and actually animal cells as well, are supported by water that moves into the cell. A plant cell has that huge vacuole where it stores the water. How does it get the water? Well, obviously it has to get the water from outside the cell. And how does it do so? By diffusion or osmosis through the cell membrane. Plants that are not watered, plants that don't have enough water, well, the plant itself will shrivel up and shrink, and the reason is because the vacuoles are no longer filled with water, causing it to have the same structure that it normally would. Organisms living in fresh water have to make sure that they don't absorb things from the outside that are not good for the cell. They have to use energy to counteract osmosis because otherwise things that are living, other organisms, things that you don't want inside your cell, would automatically flow through the membrane into the cell. So it actually has to use these proteins in the outside to keep things out. Here's an example. A toad egg cell is placed in a hypotonic solution, which means in a solution with less concentrate. It will swell via osmosis. It will get bigger via osmosis because hypotonic again means there is less solution in the inside, which means the water will flow from the outside to the inside. And to the extent that that is not good for the cell will have to use energy in order to keep it out. Osmosis, of course, also helps with the transport of nutrients from inside the cell to outside the cell. An example, bacteria, this one, Vibrio chloride, it doesn't really matter what the bacteria is, but a particular bacteria releases a toxin that blocks a channel protein in the membranes of the cell, and that's one way it keeps sodium from moving from the intestine into cells. That's just one example of a particular type of bacteria that helps human beings, that lives in a human being or an animal digestive tract, and in this case prevents sodium ions from being transported into cells. Water, as we discussed before, leaves the cell and enters the intestines through osmosis. Now this little diagram serves to illustrate what happens to cells when they go into liquids that have different concentrations. Now the ideal thing for a cell is to have the same concentration as what's outside. That's the middle diagram. The middle diagram is isotonic, where you have the same concentration of what's inside the cell to the solution that's outside the cell. And so you got the same amount of water, you got water going in, you got water going out, you got the same amount of water going in and out of the cell, and so you have no problems. A hypotonic solution on the left, this is where you have a higher concentration inside the cell than outside the cell. Now when you have a higher concentration inside the cell than outside the cell, so the water, the solution, the materials, the salt, the sugar, whatever it is, would ordinarily go out of the cell. But if the cell membrane didn't let them out, as is the case with many materials that are dissolved, the water gets drawn inside the cell. Remember, overall the water, once the concentration is going to equalize between inside the cell and outside the cell. And so what happens there is the cell is eventually going to burst, because what's going to happen, the water is going to be drawn inside the cell. The hypotonic solution, which means the outside of the cell, has less concentration than inside. So the water is going to seek the area where there's more stuff, which is inside the cell, and the cell is going to burst. What you have over here, what you have on the right side, is the opposite. You have a hypertonic solution where you put a cell, let's say, for example, in salt water. Now, the salt water is a higher concentration than the cell, and therefore the water is going to be drawn from inside the cell to outside the cell in order to equalize the concentration, and the cell is kind of going to shrivel up. This is what happens when you salt chicken and the blood gets drawn out into the salt. But if you put that's why if you put cells like bacterial cells in salt water, they're probably going to die, because the water from inside is going to be drawn outside, and the thing is going to end up shriveling up until it has not enough water inside to function, and it's probably going to die. So now let's look at some end of lesson multiple choice questions over here. And first we have question A. What would happen if the plasma membrane of the human cell or a regular cell, an animal cell, doesn't matter, were composed solely of phospholipids, which means those fat cells, those orange fat cells on the outside, and no proteins, none of those things in the red things in the diagram that open up to allow things in and out. So it's a good question. What would happen? Would it all movement across the membrane cease? No, not necessarily, because remember, water can diffuse, water can move in between the little crevices of those phospholipids, of those fatty things. Water can kind of just diffuse in and out, so that's not true. Facilitated diffusion, active transport, and osmosis would not occur. Again, not true. First of all, osmosis could happen. Active transport that requires energy could certainly happen. The cell could kind of swallow. We didn't discuss it in this chapter, but the cell can swallow what it needs to from the outside through active transport, so that's certainly not true. C, the movement of molecules across the membrane would not be affected. That's also not true, because if you wouldn't have the proteins to open up, then the cells that need to get in and out would not have as easy a time doing so. Immune reactions would not be affected. We didn't really discuss that in this chapter, but that's also not true. White blood cells would certainly be affected by not when it attacks things or tries to swallow things up, the proteins are necessary for that. E, of course, is the only one left, and why is that true? Because simple diffusion and osmosis, water moving in and out of the cell, would definitely continue to occur even if there were no proteins, because that only requires the crevices between the lipids, which it has. So we're going to choose E for this one. A cell from a wheat plant, it's a plant cell, the fact that it's wheat makes no difference, it contains all of the following except except a what? A flagellum, a cell wall, chloroplast, mitochondrion, or a cytoskeleton. Well, a flagellum is something that causes the cell to move. That's like a little tail, a little whip tail that allows it to move. Does a wheat cell, a plant cell, move? The answer, of course, is no. So A is the correct answer. A cell wall, well, that a plant cell does have that. Plant cell also has chloroplasts. All cells have mitochondrion for the respiration, and all cells have cytoskeletons, which are inside the cell that make up the materials like the membrane and other materials that the cell needs in order to have its structure. So the best answer here would clearly be A.